My name is Michael Nodine. I'm a group leader at the Gregor Mendel Institute of the Austrian Academy of Sciences.
Was ist Ihre Forschungsfrage?
In general, I'm interested in plant embryogenesis and how genes are regulated during seed development. One particular question that we're interested in is how do small RNAs, which are short RNA molecules that do not encode for proteins, regulate genes with high specificity during plant embryogenesis? Moreover, how does this small RNA-mediated gene repression affect a variety of biological processes, such as the establishment of the basic plant body plan and regulation of important phase transitions during seed development? Plant embryos are small and they contain hundreds of thousands of these small RNA molecules. So we also develop technology and software that help us analyze these populations, and these also have more general applications to both basic and applied research.
Wie finden Sie eine Antwort?
We develop and apply various technologies to profile these small and complex small RNA populations in embryos. We use Arabidopsis thaliana as a model system, a really starting point to develop these technologies and determine the best way to analyze them. For instance, we use so-called next-generation sequencing techniques where we sequence hundreds of millions of data points and then computationally analyze these to develop hypotheses about what the functions of these small RNAs may be. We then use genetics and microscopy-based approaches to test these hypotheses in a more focused manner.
Was haben Sie bisher herausgefunden?
By profiling small RNAs and their targets in developing embryos, we've discovered dozens of interactions between microRNAs and targets, including 30 targets that encode transcription factors, which can also bind to DNA and regulate gene expression themselves. Accordingly, microRNA-mediated repression of transcription factors has a profound impact on embryonic gene expression programs and is also required for the establishment of the fundamental plant body plan. We have also found that thousands of transposable elements produce exceptionally large amounts of small RNAs in plant embryos. So transposable elements are usually inert, inactive components of the genome, but can become active and mobilize and cause deleterious mutations. What we found through computational microscopy-based approaches is that as part of a feedback loop, these small RNAs are produced and help repress transposable elements and ensure that they are silenced in the new generation.
Warum ist das wichtig zu wissen?
Characterizing embryonic small RNAs has taught us a lot about how cell types are differentiated and how genes are regulated during plant embryogenesis. Moreover, using the technologies that we've produced to study embryonic small RNAs, we're addressing a wide range of questions with collaborators around the world working on both plants and animals. Moreover, using the technological know-how that we've developed with Arabidopsis, we can now apply this know-how to crops for agricultural improvement. And I think that's what's the most exciting. We're just starting to begin to understand how gene regulatory information is established in the early embryo and how this may affect the capacity of the future plant to respond to various environmental stimuli.